Hi everyone, welcome back to the Bioinformatics Coach channel. In today's video, I am going to give you 6 databases from where you can download gene expression data. Gene expression data provides us with expression profiles for genes, and thanks to high throughput sequencing technology, there are now lots of gene expression data available. These data sets are archived as microarray or RNA-seq data in their raw forms or in their processed forms. So it's up to you to know what works best for you and then just select the appropriate file or files. So let's get started. Let's switch to the browser and then we will proceed. Okay, so I'll start with the first database, which is gene expression Omnibus. We have it here. I'm going to leave all the links for the respective databases in the description box, so don't worry. So this is gene expression Omnibus, GEO for short. And so you can see a brief description here. So you can use this to read about the database. So here it says that it's a public functional genomics data repository. Okay. And so you can have data sets here for your studies. Now with the gene expression Omnibus, there is a search box here which you can use to search for keywords or accession IDs, accession numbers, whatever you want to call it. So let's just say you want to search for this study. It has this accession number gene SE129960. So once you put it here, you just search, and then it will return the hit for you, assuming that the ID that you selected is valid. Okay, so when you are here, you get information about the data. You can have status, title, organism, which is important, experiments, type, summary, overall design, and which can also be the title to a publication. We have contributors, and we also have the contacts, the contact person, okay, or corresponding author. So here you just look at them, and then you can even send a mail or contact this person and then make inquiries. I remember I was working on a gene expression data analysis work, and then I needed some information about the data that I had seen here. And so what I did was to contact the person and then just made the inquiry. So you can also do that here. You can see the platforms also here. The platforms will let you know what sequencing platform was used. And then we also have the samples. You can just click more. Let me just go back here. So here you have less, more. You have the number of samples here, and then here you have files that you can download. There's also this one here, the supplementary file. So this supplementary file could be the read count, or it could be other files which were generated, okay, from the original data. So that's it. And then we have some links here to lead you to the data itself. So for this tutorial, it's just an intro, so I will not go into details with all these databases, so I will skip them. Now let's go back to the home page here. So with GEO, you have this getting started section here to give you an overview. You can also look at the frequently asked questions about data sets, profiles, etc. And there are tools here to also help you to make queries. So the tools here can help you to download the data, can help you to search, can also help you to analyze the data. So let's look at them here. We have tools for searching studies, tools for searching for gene expression profiles. We also have those for documentation, and we also have tools to help you to analyze a study. So this one here, the tool for analyzing a study is GEO2R. So here you can use your browser to do the analysis, and then what happens is that behind the scenes R commands will be executed and then that will help you to do the analysis. And you can also download the R scripts after the studies after the analysis has been done. Okay, so these are things you can do with gene expression Omnibus. You can also browse the contents. We have data sets, series platform, etc. So it's up to you to search and then just get what you want. Now let's proceed. We will move on to the next database, and that is SRA. Okay, so SRA, that is Sequence Read Archive, also has data sets. Okay, so this repository has high throughput sequencing data. So it's not only gene expression data you can find here. You can also find whole genome sequence, whole exome sequence data as well. So just search for them. But for this talk, it's about gene expression data. So that's what we are going to focus on. So let's just look at this. We can search here with the SRA archive. There is a search box here for you to search keywords or ID. So let's just search for an ID. Let's search for this SRAToolkit57630. So let's search for this. So this is a data. Okay, so when you have the accession ID or accession number and you search, you have information about that data. Okay, so you have them here. You have library, instrument, strategy, source, selection, layout. The layout is also necessary if you are interested in downloading FASTQ reads. Then you need to know whether it's a paired end read or single end. So the layout will tell you that. And then you can also proceed to download the data. So let's search. You can just use this link, and then when you click on it, it will send you to another page, and then from there you can also download. Okay, you can download. Let's just check. Let's search here. Okay, yeah, so you can download using this button here. You can just check it out. Okay, so it's just up to you to explore. So I'll not go into details with that. Let's just check if there's something else here. Okay, I think that's about it. So there's just a brief intro to the SRA archive or the SRA repository. With the SRA, you can also do advanced queries, advanced search using this button here. Okay, this the advanced button. So just click it, and then you have this coming up, and then you can search, and then even select fields, and then this will help you to narrow down to what you really want or what you want. So it's very useful. Okay, so let's go back. Yeah, with the SRA repository, there are two ways that you can use to download data. You can use the browser, the web interface here, or you can also use the toolkit. What we've been doing so far is to use the browser to search and then download data, but you can also use the command line to help you download it. And I've covered 2 tutorials on that. As at the time of making this video, I had covered 2 tutorials. So one shows you how to download FASTQ reads. One also shows you how to download FASTA files. So you can just watch those videos. The links to those videos are in the description box. The command line tool is very useful if you have lots of data that you want to download, or if you want to include the data download activity in a pipeline, then it becomes very, very, very handy. Okay, but if you have just a few samples to download, or just a small number of samples or files to download, then it's better to just use a browser to download it. So there are pros and cons, okay, when using the command line tool or the browser to download the data. So you just need to look at what works best for you and what is more convenient for you. So that's about it. So let's move on to the third database, which is Array Express. So with the databases, you can also search for information. Okay, you can just check the some of the tabs which will link you to a brief description of the database. So for Array Express, for example, there's just about Array Express here, which tells us more. So when you click on it, it tells you what is Array Express. So you have some notes here to help you know what this database is about. So from this page here, we can see that it's one of the repositories for recommended by scientific journals to archive functional genomics data. Okay, so that's about it. Now with Array Express, let's go back to the home page. With Array Express, you can search, okay, maybe a keyword here, let's say cancer, or you can even search using accession IDs like this one here, and then once you search, you have the result coming up. Let's just search for cancer. So you have some results coming up, but you can also use this search button here to search for the term. Okay, so that's about it. Now with Array Express, there is an advanced search button here. Okay, that is used for making advanced search. So that is also here. So you just click on the advanced search here, and then you have this page coming up that tells you how to perform the advanced search. So it's a very cool platform. You can also browse the repository. Just use this button here, browse, and then once you click on it, you have the entries being given for you. So you have a table, accession ID, title, and then type of data, RNA-seq, ChIP-seq, etc. Organism is also there, assays, released, process raw, etc. So just look at what works best for you. Okay, so that's about it. You can also submit data, but for this tutorial, we are looking at how to download data, so I will not go into this particular one. Okay, so let's move on to the fourth database, which is Expression Atlas. Expression Atlas, again, we can check using the About button here, and then we will get some short notes, okay, telling us what this database is. So here it says it's an open science resource that gives users a powerful way to find information about gene and protein expression. So at least we know what it's about. So with Expression Atlas, there is a search box here that you can use to search for gene and then proteins, and then you can also limit to the species, or you can use all the species. That's any of the species. So you can just select one, and then that will help you to narrow your search results. You can also include biological conditions. If you check here, it says that you can search across 65 species, over 4,000 studies, over 150,000 assays. So this also lets you know, okay, what results you should expect. You can search, and then you can also browse through this database. You can search for information about Homo sapiens. You can look at studies or experiments that use Homo sapiens samples. You can also search most musculus. There are a host of organisms here, so you just need to look at the organism of interest and then just search. You can look at plants, fungi. So there are a lot of them here, so you just search and then look at what works best for you. You can also search for single cell experiments. Single cell is becoming very popular, and so it makes sense to add this functionality. So you can just come here to search for it, just click, and then you can now search here. So that's about it. You can also go back to the Expression Atlas and then start from there. So this is it for the Expression Atlas. So I encourage you to search, just play around with it, and then see what comes up. Let's look at database number 5, and that is the TCGA program. Okay, so TCGA, that is the Cancer Genome Atlas program. This is a database that has genomic data for cancer studies. So over the years, there has been lots of data collected. Okay, so if you are interested in cancer studies, you want to get gene expression data for cancer studies, then it's good to come to this database, because this database has lots of data for you. I myself have used this database a couple of times, so it's cool. So here, if you want to get the data, just scroll down and then come to access TCGA data. So just click it, and then you'll be sent to this page. So here you can look at projects, you can explore, you can analyze, but our focus is downloads, so we will use the repository. So just click the repository, and then you have this coming up. And so here you can filter. Okay, this will help you to narrow down the results. Since it's gene expression data, we have to select transcriptome profile, and then you can proceed to also check. Okay, if you want single cell analysis, you want gene expression quantification, you can just check them and then just look at what works best for you. And by the way, I have already covered a separate tutorial that shows you how to download gene expression data from the TCGA database, so you can just watch that video. The link is in the description box, so just do that. I think I've covered two. One uses the browser, and then one uses Python. So just look at which of those videos works for you, and then just learn from them. So that's about it. So the TCGA database is not just about gene expression. There are other data sets as well, so you can just search for them. So that's about it. Now let's go back. Let's search. Yeah, we can go back here. Okay, so let's check. Okay, so with the TCGA, you can search using files, or you can also search using cases. I forgot to mention that. So the cases are here. So just click the cases, and then here you can look at the programs or studies, and then select the one which is okay for you. So that's about it. Now let's look at the last one, which is the Detex database, or genotype tissue expression program. Okay, so this database also has data for human samples. So here, if you want to get to the database, just come to the Detex portal. This side here, there's this Detex portal. I'll leave the link in the description box, by the way. So just click it, and then you'll be sent to the portal. So here you can search using the gene or the SNP ID, or you can also search by tissue, histology. There are lots of things here, so you just need to look at what works best for you. You can come to downloads, and then check for open access data or protected access data. Now this brings me to this particular point. With biological databases, you have data sets that are publicly available, and there are those that are restricted. But most of the databases are, most of the data sets are publicly available. Okay, some are restricted. So you can search and then look at the ones that work for you. If it's open access, it's free to download, it's accessible. But if it's restricted, then it could be that you need to be in a particular consortium before you can access the data, or you need to apply, or you need to follow some procedure before you can get access to the data. So even though it's protected or access is restricted, you can just look at the documentation and see if there's a way to get that data or not. So just check and then get it. And that is why it's important to also read the documentation. So with the Detex, there is a documentation tab here that will give you information about Detex, and then other stuffs. And then here we also have current release. So I'll use this point here to explain something. These databases are regularly updated, and some of them will give you versions. And so what happens is that you can have some information available in one version but absent in another, or that information may be modified. Okay, so you need to look at the versions and then decide if this version works for you or not. It's very, very important. Okay, this is important if you want to reproduce a study, so take note of that. So that's about it. Now we've covered 6 databases, so I think so far so good. Now let's proceed. So this brings me to the end of this tutorial, and before I go, let me also say that I already have some tutorials that show you how to perform gene expression analysis, so you can just watch them. The links to those videos are in the description box, so just try and watch them if you want to learn how to analyze RNA-seq data from scratch, from the raw reads. Okay, then you can watch those videos to help you get started. If there are databases that you also know, you can just put them in the description box. This list I made mention of, or this list of databases I showed, is a non-exhaustive one, meaning there are other databases available, and so just try and then put the others in the comment section so that we can all have some discussions going on. So that'll be all for this tutorial. Thanks for watching, and I'll also see you in the next session. Goodbye.